Hey guys, welcome to my YouTube channel. In this video, we're going to be going through the cell cycle. Now, what is the cell cycle and why is it important? So, the cell cycle is a series of steps or phases that a cell needs to go through in order to produce two identical donor cells. And we need new cells all the time. For example, we constantly shed skin cells that need to be replaced, and we also need new cells for growth and repair. The cell cycle is divided into two main phases, interphase and mitosis. Interphase is a longer phase where the cell prepares to divide, and this is divided into G1, S, and G2 phases. Mitosis is a phase that follows. This is divided into PMAT, Prophase, Metaphase, Anaphase, and Telophase, and this is where the cell actually divides. Now, let's start looking at interphase. So, this is the copy of the notes that I'll be using for this tutorial, and as I promised, I will provide a free copy. You can find the link in the description box below, so feel free to download. However, just make sure that you hit the like and subscribe button on your way. Before we go ahead, let me introduce the concept of checkpoints in the cell cycle. I will be covering checkpoints in a lot more detail in my next video about the regulation of the cell cycle, but basically, checkpoints are sort of a self-check to check the integrity of the cell's genome. And what is the genome? The genome is a collection of all the genetic information of a cell, i.e. all the DNA in the cell. And the whole point of this is to prevent the proliferation of mutant cells. Now, mutant cells are cells with a damaged genome, and preventing the proliferation of mutant cells is crucial because DNA needs to be preserved between replication cycles. Now, if we allow these mutant cells to progress through the cell cycle and divide, then we will be causing the accumulation of mutations. And this will not only reduce the functioning capacity of cells, but could also lead to cancer. So, if everything's okay, then the cell will progress through the cell cycle. However, if there is damage detected, then first of all, repair will be attempted, and if repair is not possible, then the cell will undergo apoptosis, which is programmed cell death. And once again, this is because we don't want mutant cells to be proliferating. Now, let's get back to interphase. So, this is the longest phase. It's about 12 to 24 hours long, and it's divided into G1, S, and G2 as I mentioned earlier. Now, it's important to note that G1 and G2 are also known as gap 1 and gap 2. I like to think of G1 and G2 phases as growth phases in which the cells are growing as preparation for the next phase. So, the first phase in interphase is the G1 phase. It's roughly 10 hours long, and this is where the cell decides if it needs to divide. Now, this decision is made by the presence or the absence of growth factors. If growth factors are present, then they signal the cell to divide, and so the cell grows in size and doubles the number of organelles, and then eventually moves on to the S phase. On the other hand, if growth factors are absent, then the cell will exit the cell cycle and move out to the G0 phase. The G0 phase is not part of the cell cycle, and it's sort of a resting phase where the cells are not dividing. Now, cells can leave the cell cycle temporarily. For example, stem cells, which can re-enter the cell cycle quite easily whenever cells are needed. Cells can also leave the cell cycle permanently, and this can happen for two main reasons. Firstly, if the cells are terminally differentiated. This means that they don't need to divide any further, and a good example are neurons. The second reason is if the cells have reached the Hayflick limit, meaning that the cells are senesced. The Hayflick limit is a set number of times that a cell can divide. And this is another defense mechanism against cancers. Anyways, let's get back to the cell cycle. Now, assuming that growth factors are present, the cell needs to go through two cell cycle checkpoints. The first one is a restriction checkpoint, which is sensitive to cell size, and this ensures that the cell is the correct size for division. And secondly, we have our first DNA damage checkpoint, known as the G1 DNA damage checkpoint. Once both the checkpoints are passed, the cell can move on to the S phase. This is where DNA synthesis takes place and the DNA content doubles. The DNA content in a cell is normally 2N, N referring to 23 chromosomes and the 2 meaning 23 pairs of chromosomes. However, after the S phase, we have 4N, and this is because each chromosome becomes a pair of sister chromatids joined by a centromere. Doubling the DNA content takes about 4 to 6 hours, and once the DNA content is doubled, the cell can move on to the G2 phase. But before moving on to the gap 2 phase or the G2 phase, the cell needs to go through another DNA damage checkpoint known as the intra-S phase checkpoint. The G2 phase is the last phase of interphase. It's about 4 hours long, and this is where the cell does some final preparations before entering mitosis. In this phase, cell growth and protein synthesis take place, followed by the third DNA damage checkpoint, known as the G2 DNA damage checkpoint. The integrity of the genome is crucial, and this is demonstrated by the presence of not one, not two, but 3 DNA damage checkpoints. The second major phase of cell division is mitosis. Now, this phase is a short phase. It's about 1 to 2 hours long, and this is where the physical cell division takes place. Now, growth and protein production stops, and all the cell's energy is focused on orderly division into 2 identical daughter cells from a single cell. Before we go ahead, let's clarify some potentially confusing terms. So, first of all, there's chromatin. This is uncoiled DNA that is found in normal cells that are not dividing. And during cell division, this condenses down into chromosomes. Chromosomes are these X-shaped structures that are made of 2 identical sister chromatids. So basically, each one of these is an exact copy that is joined together at the center by a structure known as centromere. Another structure that sounds similar is centrosomes. These are organelles that are found near the nucleus, and they form microtubules known as spindle fibers. Next, we have the nucleus versus the nucleolus. The nucleus is the region which contains all the DNA, and the nucleolus is a dense region within the nucleus that is responsible for the production of ribosomal RNA. So, nucleolus, production of ribosomal RNA. So, let's get back to mitosis. Mitosis is made up of 4 main phases: prophase, metaphase, anaphase, and telophase. And a nice way to remember this is PMAT. P-M-A-T. PMAT. And following these 4 phases, we have cytokinesis. So let's get into the details. The first phase of mitosis is the prophase, and there's 4 main changes that take place. Firstly, the chromatin, the uncoiled DNA, condenses down into chromosomes. Secondly, the nucleolus disappears. Remember, the nucleolus disappears, not the nucleus. The nuclear membrane is still intact. Thirdly, the centrosomes start separating. They start moving towards opposite poles. And the centrosomes start forming spindle fibers. These spindle fibers will attach to centromeres, and it will eventually pull the sister chromatids apart. The next phase is metaphase. However, some books mention another phase that is between prophase and metaphase, and this is known as the prometaphase. The most noteworthy changes that take place in this phase are that the nuclear envelope breaks down. However, you can still see some fragments of the nuclear membrane, and the centromeres have reached opposite poles. Furthermore, some microtubules attach to centromeres, and the chromosomes have started to line up in the center. Let's move into metaphase. Now here, the nucleus has completely disappeared, and all the chromosomes have nicely lined up at the center, forming the equatorial or metaphase plate that you can clearly see in this histology image. Spindle fibers from both centrosomes have attached to the chromosomes via the centromeres and are ready to pull them apart. It is important to note that there is another checkpoint at metaphase, and this is the 5th checkpoint. It is known as the spindle assembly checkpoint, and it ensures that the chromosomal segregation is even, which means that each of the 2 daughter cells will receive the same number of chromosomes. Now, the next phase is anaphase, and here the spindle fibers start shortening and pulling the chromosomes apart. The centromeres have broken apart, and on histology, you can see these V-shaped chromosomes moving towards opposite poles. Once the chromosomes are separated, the cell will enter telophase. Here, the chromosomes will decondense at opposite poles, and nuclear envelopes will reform. Finally, the cell membrane separates to form 2 identical daughter cells, and this process is known as cytokinesis. Here, microtubules reorganize into cytoskeleton, and the 2 cells return to the G1 phase of interphase, where the presence or the absence of growth factors will decide whether the cells will exit the cell cycle or divide again. So, this is the end of the video. Thank you so much for watching. If this video was helpful, please hit the like button and make sure to subscribe for more tutorials. Also, please make sure to share this video with anyone who will find this video useful. It just takes a few clicks. Make sure you leave any questions or suggestions in the comment box below. This is the Caffeinated Medic signing off. Till next time, take care.